Picture yourself settling into a comfortable space, your breathing becoming slower and deeper with each passing moment, your muscles relaxing, releasing the tension accumulated throughout the day. Tonight, we're embarking on a journey through the most extraordinary structure in the known universe, the human brain. This 3-pound marvel sitting within your skull right now, cradled safely within protective bone and cushioning fluid, contains more connections than there are stars in the Milky Way galaxy, more complexity than any supercomputer ever built, and more mysteries than we may ever fully unravel. Every thought you've ever had, every emotion you've ever felt, every memory you've ever stored, all of it emerges from this remarkable organ, this biological miracle of interconnected neurons and glial cells, of chemical signals and electrical impulses, of structure and function dancing together in perfect coordination. As you relax and let your mind drift, feeling your body becoming heavier, sinking deeper into comfort with each breath, we'll explore 100 fascinating facts about this incredible organ. Each discovery reveals another layer of the beautiful complexity that makes you who you are, another window into the mechanics of consciousness itself. Some of these facts will surprise you, others will inspire wonder, and all of them will deepen your appreciation for the extraordinary machine operating silently inside your head every moment of every day. Let the gentle rhythm of these discoveries carry you into a peaceful sleep, where your own brain will continue its nightly work of consolidation, restoration, and wonder. Your brain never stops working for you, even in the deepest sleep, organizing memories, clearing waste, dreaming dreams, preparing you for tomorrow. Close your eyes now, breathe deeply, and let's begin this calming exploration of the universe within your head. Section 1. The brain's architecture. Building blocks of consciousness. Fact number 1 takes us to the very foundation of who we are. Your brain weighs approximately 3 pounds, about the same as a half-gallon of milk sitting in your refrigerator, yet it commands your entire existence. Despite representing only about 2% of your total body weight, this modest mass commands an extraordinary 20% of your body's total oxygen supply and 25% of its glucose. Think about that for a moment. 1/5 of every breath you take, 1/4 of every meal you eat goes to feed this hungry organ. Every moment you exist, every thought you think, every memory you form, every decision you make requires this constant flow of energy, making your brain one of the most metabolically demanding organs in your entire body. The neurons in your brain burn through glucose at a rate that would be unsustainable for any other tissue, yet they never tire, never rest, never stop their essential work of generating your conscious experience. The heaviest normal human brain ever recorded weighed 4.43 pounds and belonged to the Russian writer Ivan Turgenev, a man known for his literary genius, his passionate advocacy for social reform, and his complex emotional life. Though, interestingly, brain size doesn't correlate with intelligence, a reminder that complexity and capability far outweigh simple measurements of mass. Einstein's brain, famously preserved and studied after his death, weighed only 1,250 grams, slightly below average for an adult male. What mattered wasn't the size, but the organization, the density of connections, the efficiency of networks. Your brain's power lies not in its weight, but in its wiring, not in its mass, but in the miraculous dance of electrochemical signals flowing through its intricate pathways every millisecond of your life. Fact number 2 reveals the texture of thought itself. If you could hold a human brain in your hands, carefully, respectfully, with full awareness of the profound life it once sustained, it would feel like firm jelly, a consistency that seems almost impossibly delicate for something so powerful. The tissue would yield slightly to gentle pressure, like a ripe avocado or perfectly set custard, yet hold together with surprising cohesion. This soft, pinkish-grey tissue, protected by the hard shell of your skull and cushioned by cerebrospinal fluid that surrounds it like a protective water bath, somehow generates every emotion you've ever felt, every decision you've ever made, every dream you've ever experienced, every spark of creativity, every moment of joy or sorrow or wonder.
The delicate nature of brain tissue, more fragile than you might imagine for something so essential, is why evolution invested so heavily in protecting it, surrounding it with multiple layers of defense. First, the skull, that hard fortress of bone. Then the meninges, 3 layers of membrane wrapped around the brain like protective blankets. Then the cerebrospinal fluid, that clear liquid cushion that surrounds the brain, allowing it to float weightlessly, protected from the jarring impacts that could damage such tender tissue if it rested directly against bone. Finally, the blood-brain barrier, that selective filter that keeps harmful substances from entering while allowing necessary nutrients through. All these protections exist for one purpose: to shelter the most precious cargo imaginable. The 3-pound universe that makes you conscious, makes you human, makes you you. Our third fact takes us deep into the numbers that make up your mind. Your brain contains approximately 86 billion neurons, those specialized cells that process and transmit information through electrical and chemical signals. To put this number in perspective, if you tried to count each neuron at a rate of 1 per second, it would take you over 2,700 years to count them all. These neurons aren't simple switches, but intricate cellular machines, each one a living entity with its own metabolic needs, its own complex internal machinery, and its own unique role in the vast network of your consciousness. Fact number 4 explores the connections that bind these neurons together. Each of those 86 billion neurons forms connections with thousands of other neurons, creating what scientists estimate to be somewhere between 100 trillion and 1 quadrillion synapses, the tiny gaps where neurons communicate with each other. A quadrillion is a 1 followed by 15 zeros, a number so vast that if you could examine 1 synapse every second, you'd need over 30 million years to look at them all. These synaptic connections form and break and strengthen and weaken throughout your entire life, creating the ever-changing landscape of your thoughts, memories, and capabilities. Our 5th fact reveals a startling truth about the composition of your brain. 60% of your brain is made of fat, making it the fattiest organ in your entire body. But this isn't the kind of fat that collects around your waistline. These are specialized fatty acids, particularly omega-3 fatty acids like DHA, that form the myelin sheaths wrapping around nerve fibers and make up the structural components of brain cell membranes. Every cell membrane in your brain depends on these fats for its integrity. Every electrical signal traveling through your nervous system relies on them for speed and accuracy. When you consume foods rich in healthy fats, fish, nuts, avocados, you're literally feeding the structural foundation of your thoughts. Fact number 6 takes us to a remarkable discovery about the brain's organization. The cerebrum, the large, wrinkled outer portion of your brain that you probably picture when you think of the brain, makes up 85% of your brain's total weight. This massive structure divides into two hemispheres, left and right, connected by a thick bundle of nerve fibers called the corpus callosum that contains approximately 200 million axons. These hemispheres work together in a sophisticated dance of specialization and integration, with the left side of your brain primarily controlling the right side of your body and vice versa, a crossover that occurs in the brainstem and remains one of the curious features of our neurological architecture. Our seventh fact explores the wrinkled surface that gives the brain its distinctive appearance. The cerebral cortex, that thin layer of tissue covering the outside of your cerebrum, is only about 2 to 3 millimeters thick, roughly the thickness of a stack of 2 or 3 dimes. Yet, despite its thinness, this is where most of the actual information processing in your brain takes place. If you could unfold and flatten out this cortex, it would cover an area of about 2,500 square centimeters, approximately the size of a large dinner napkin. The brain's solution to fitting this much surface area inside your skull was to fold it up into those characteristic ridges called gyri and grooves called sulci, maximizing the processing area while minimizing the volume.
Fact number 8 reveals the color behind the common phrase, gray matter. The cerebral cortex appears gray because the nerve cells in this area lack myelin insulation, that fatty white coating that makes other parts of the brain appear white. When people talk about using their gray matter, they're referring to this thin layer where conscious thought, decision-making, and higher-order processing occur. The white matter lies deeper in the brain, composed of myelinated axons that form the communication highways connecting different regions of gray matter to each other, enabling the integrated function that characterizes human cognition. Our ninth fact takes us to a remarkable property of brain tissue. A piece of brain tissue the size of a grain of sand contains approximately 100,000 neurons and 1 billion synapses. Imagine holding a single grain of sand between your fingers. Within a similarly sized chunk of your brain exists a neural network of staggering complexity, with enough connections to process multiple streams of information simultaneously. This density of processing power, packed into such a small volume, exceeds anything that current technology can replicate, making every grain-sized piece of your brain more computationally sophisticated than the most advanced computer chips ever designed. Fact number 10 addresses one of the most persistent myths about the brain. You've probably heard the claim that humans only use 10% of their brains, but neurologists have thoroughly debunked this fiction. Brain imaging studies show that you use virtually all of your brain, even when you're sleeping. Different areas activate for different tasks, and there's certainly variation in activity levels throughout the day, but there's no silent 90% waiting to be unlocked. Even during rest, your brain buzzes with activity, maintaining body functions, consolidating memories, processing emotions, and running simulations that neuroscientists are only beginning to understand. Your brain is always on, always working, always consuming those precious resources we mentioned earlier, because consciousness and life itself depend on this constant whole brain engagement. Fact number 11 brings us to the speed of thought itself. When a neuron fires, it generates an electrical impulse called an action potential that can travel along nerve fibers at speeds of up to 270 miles per hour. That's 350 kilometers per hour, faster than the fastest Formula One race cars. The signals traveling from your brain throughout your body, instructing your muscles to move, telling your organs how to function, bringing sensory information back to consciousness, zip along these biological wires at astonishing velocities. Some of the slowest nerve signals, like those involved in pain perception from unmyelinated fibers, still travel at about 2 miles per hour, while the fastest can reach those incredible peak velocities, turning your body into a telecommunications network of extraordinary speed and sophistication. Our 12th fact explores the mysterious moment of neural communication. When an action potential reaches the end of a neuron's axon, it triggers the release of chemical messengers called neurotransmitters into the synaptic gap, that microscopic space between neurons measuring only about 20 to 40 nanometers across, roughly 1,000th the width of a human hair. These neurotransmitter molecules float across this tiny gap, taking less than a millisecond to traverse the distance, and bind to receptor proteins on the receiving neuron. This chemical synaptic transmission, occurring at trillions of synapses throughout your brain every second, translates the electrical language of one neuron into chemical signals that can either excite or inhibit the next neuron in the chain, creating the complex symphony of brain activity that underlies everything you experience. Fact number 13 reveals the power requirements of consciousness. Your brain operates on approximately 20 watts of power, about the same as a dim incandescent light bulb. This remarkably efficient organ, running on less power than most smartphones, generates all of human experience, creativity, emotion, and thought. By comparison, the most powerful supercomputers require millions of watts to perform calculations that still can't match what your 20-watt brain does effortlessly every moment. Recognizing faces, understanding language, feeling emotions, generating new ideas, navigating complex social situations, and maintaining consciousness itself. Our 14th fact takes us to a peculiar phenomenon you've likely experienced. When you eat or drink something very cold too quickly, you might experience a brain freeze, medically known as sphenopalatine ganglioneuralgia, a delightfully specific term for that brief, intense headache. This occurs
When the cold hits receptors in the outer covering of the brain called the meninges, causing rapid constriction and then dilation of blood vessels in that area. The sudden change in blood vessel diameter triggers pain receptors, creating that characteristic sharp pain that typically subsides within 30 to 60 seconds. The brain tissue itself, interestingly, cannot feel pain, which leads us to our next fact. Fact number 15 addresses a curious truth about pain and the brain. Despite being the organ responsible for processing all the pain signals from your body, the brain itself contains no pain receptors. Neurosurgeons can operate on exposed brain tissue while a patient is awake, and the patient feels no pain from the brain tissue being touched, moved, or even cut. The pain we experience during headaches comes not from the brain tissue, but from the meninges, the blood vessels surrounding the brain, the periosteum covering the bones of the skull, and the muscles and skin of the head and neck, all of which contain abundant pain receptors. This strange irony means that the organ that makes you conscious of all bodily pain is itself incapable of experiencing pain directly. Our 16th fact explores a surprising aspect of brain cell populations. Neurons aren't the only cells in your brain. In fact, they're outnumbered by supporting cells called glia, derived from the Greek word for glue. For decades, scientists thought these glial cells simply held neurons in place and provided structural support. But modern research reveals they play active roles in brain function. They help regulate the chemical environment around neurons, form the myelin insulation around nerve fibers, remove dead cells and debris, assist in forming new synaptic connections, and may even participate directly in information processing. Some estimates suggest you have 10 times as many glial cells as neurons, though more recent counts put the ratio closer to 1:1, still representing tens of billions of these crucial supporting cells working silently to keep your brain functioning. Fact number 17 brings us to the brain's remarkable isolation from the rest of the body. The blood-brain barrier, a selective membrane formed by specialized cells lining your brain's blood vessels, carefully controls what substances can pass from your bloodstream into your brain tissue. This barrier protects your brain from toxins, pathogens, and random fluctuations in blood chemistry that might disrupt delicate neural function. Only specific molecules, oxygen, glucose, certain amino acids, and water, can pass freely through this barrier, while it blocks many medications, toxins, and even some nutrients. This protection comes with trade-offs, making it challenging to deliver drugs to treat brain diseases, and meaning that the brain must manufacture many of its own specialized molecules, including all of its cholesterol, since cholesterol from the bloodstream cannot cross this barrier. Our 18th fact reveals a startling truth about consciousness and computation. Scientists estimate that your brain can perform approximately 10 quadrillion operations per second. That's 10,000 trillion calculations occurring simultaneously throughout your neural networks right now as you listen to these words. No computer yet built can match this raw computational power, despite decades of advancement in silicon-based processing. Your brain achieves this through massive parallelism, with billions of neurons operating simultaneously rather than sequentially, each one a tiny processor in the vast distributed network of consciousness. And it accomplishes all of this while consuming just 20 watts and occupying about 1.4 liters of volume, a density and efficiency that represents the pinnacle of biological engineering. Fact number 19 explores the timing of neural processing. When you look at something, the visual information travels from your retina through your optic nerve to the back of your brain, undergoes processing in visual cortex areas, gets integrated with information from memory and other sensory systems, and becomes a conscious perception, all in about 1/10 of a second. This means you're always experiencing the world with a slight delay, seeing events that technically happened 100 milliseconds ago. Your brain works hard to hide this delay from you, predicting where moving objects will be, filling in gaps in sensory information, and creating the seamless experience of real-time perception. When you catch a ball, you're not catching where it is, but where your brain predicts it will be by the time your hand gets there, demonstrating the sophisticated predictive algorithms running constantly in your neural networks.
Our 20th fact brings us to the phenomenon of neural plasticity at the synaptic level. When you learn something new or practice a skill, the synaptic connections between relevant neurons strengthen through
A process called long-term potentiation.
neurons that fire together, wire together, in the famous phrase coined by psychologist Donald Hebb. The more frequently a particular neural pathway activates, the stronger and more efficient those connections become. Conversely, synaptic connections that go unused gradually weaken and may eventually disappear altogether, following the principle that if you don't use it, you lose it. This constant remodeling of synaptic strength, occurring throughout your life based on your experiences and behaviors, makes your brain a dynamic, ever-changing organ, continuously sculpted by the life you lead. Section 3. Memory and learning, the brain's storage system. Fact number 21 takes us deep into the temporal lobes to discover the hippocampus, that small seahorse-shaped structure from which it gets its name, crucial for forming new memories. You have 2 hippocampi, one on each side of your brain, each about 5 centimeters long. Despite their small size, these structures play an outsized role in converting experiences into long-term memories. The famous case of patient Henry Molaison, known as HM, who had both hippocampi surgically removed to treat severe epilepsy, revealed the critical importance of these structures. After surgery, HM could form no new declarative memories of facts or events, though he could still learn new motor skills, demonstrating that different types of memory depend on different brain systems. He lived in a permanent present, unable to transfer new experiences into long-term storage, teaching neuroscientists invaluable lessons about how memory works. Our 22nd fact explores the astonishing capacity of human memory. Research suggests your brain's storage capacity is virtually unlimited. There's no known upper bound to how much information you can potentially store. If we tried to quantify it in computer terms, estimates range up to 1 petabyte or more. That's 1 million gigabytes, enough to store approximately 4.5 million high-definition movies. But this comparison doesn't quite capture how memory works, because your brain doesn't store memories like a computer stores files. Instead, memories distribute across neural networks, with each memory encoded in patterns of synaptic connections. The same neurons can participate in encoding many different memories, and retrieving a memory means reactivating those patterns, not reading a file from a storage location. This distributed, pattern-based storage makes your memory both incredibly vast and fundamentally different from any artificial storage system. Fact number 23 reveals a crucial insight about memory formation. Memories are not formed instantly in their permanent state. When you experience something new, a memory trace first forms in the hippocampus, while simultaneously being encoded in the cortex. Groundbreaking research from MIT showed that these memory traces actually form simultaneously in both locations, but the cortical trace remains silent for about 2 weeks, only becoming functional after a maturation process. During sleep, especially during slow-wave sleep, the brain replays and strengthens these neural patterns, gradually transferring the memories from temporary hippocampal storage to more permanent cortical storage, a process called consolidation. This is why a good night's sleep after learning something new helps cement that information in your long-term memory. Our 24th fact takes us to different types of memory systems in your brain. Episodic memory stores your personal experiences, the specific events of your life that you can consciously recall and mentally travel back to. Your last birthday party, your first kiss, what you ate for dinner last Tuesday. Semantic memory holds general knowledge and facts about the world, things you know but don't remember learning. That Paris is the capital of France, that water freezes at 0 degrees Celsius, what a dog is. Procedural memory encompasses skills and habits, the kind of knowing that shows itself in doing rather than thinking. Riding a bicycle, typing on a keyboard, brushing your teeth. Each type relies on different neural networks, which is why someone with hippocampal damage might lose the ability to form new episodic memories while still being able to learn new skills. Or why someone with Alzheimer's disease might forget what they had for breakfast, but still remember how to play piano. Fact number 25 explores the surprising flexibility of memory. Every time you retrieve a memory, you don't just recall it, you reconstruct it. Your brain pieces together information from distributed storage sites, fills in missing details based on expectations and existing knowledge, and actually modifies the memory in subtle ways during the retrieval process itself. This means that memories aren't fixed recordings, but dynamic reconstructions, susceptible to distortion, enhancement, and even an outright fabrication. Studies show that false memories can feel just as vivid and real as accurate ones, and that merely imagining an event or being exposed to misleading information can sometimes create memories of things that never happened. Your memory serves you well most of the time, but it's more like a constantly edited and revised Wikipedia page than a photograph or video recording. Our 26th fact reveals the extraordinary phenomenon of neuroplasticity, the brain's ability to change its structure and function throughout life. Once, scientists believed the adult brain was essentially fixed, incapable of growing new neurons or making significant structural changes. Research in the latter half of the 20th century overturned this dogma, showing that the brain remains plastic throughout the lifespan. London taxi drivers, who must memorize the complex street network of the entire city, show measurable enlargement of their posterior hippocampi compared to control subjects, and this enlargement correlates with years of experience. When people lose a limb, the cortical area formerly devoted to that limb gets recruited for other purposes. Blind individuals show enhanced auditory and tactile processing in their visual cortex. Your brain continuously rewires itself based on experience, learning, and injury, maintaining the capacity for change from birth until death. Fact number 27 takes us to a discovery that challenged long-held scientific assumptions. In the 1990s, researchers discovered that the adult human brain can generate new neurons, a process called neurogenesis. This primarily occurs in the dentate gyrus of the hippocampus and the olfactory bulb. For decades, scientists had insisted that humans were born with all the neurons they would ever have, and that neurogenesis ended in early childhood. The discovery of adult neurogenesis, confirmed through multiple lines of evidence, including the presence of neural precursor cells in adult brains, opened new avenues for understanding learning, memory, and potentially treating neurodegenerative diseases. Some studies suggest that neurogenesis continues even into the 7th and 8th decades of life, though it does decline with age, and factors like exercise, learning, and environmental enrichment can enhance this natural process of neural regeneration. Our 28th fact explores the critical periods in brain development. Your brain doesn't finish developing until you're in your mid to late 20s. Brain development proceeds from back to front, with the prefrontal cortex, responsible for planning, decision-making, impulse control, and reasoning, being the last region to fully mature. This explains why teenagers and young adults sometimes make decisions that seem impulsive or poorly thought through. The neural circuitry for executive function is literally still under construction. The brain also exhibits special windows of heightened plasticity during development, called critical periods, when particular types of learning occur most easily. Language acquisition, for instance, comes much more naturally to young children than to adults, a difference attributed at least partly to this greater plasticity in the developing brain. Fact number 29 reveals a fascinating finding about different memory stages in life. Research shows that the hippocampus in one-year-old infants is already capable of forming memories, yet we adults have no conscious recollection of our first year of life or even our first few years. This phenomenon, called childhood amnesia, occurs not because these early memories were never formed, but because we can't access them later. Scientists theorize this might relate to the rapid changes occurring in the developing brain during early childhood, or to the way memories get encoded before language develops, or to the massive neurogenesis occurring in the young hippocampus, potentially disrupting old memory traces. Whatever the mechanism, the result is that your earliest experiences, though they surely shaped you, remain permanently beyond the reach of conscious recollection. Our 30th fact brings us to the discovery of different learning systems in the brain. In addition to the hippocampus-dependent explicit memory system, your brain contains implicit learning systems that operate below the level of consciousness. The basal ganglia, a group of structures deep in the brain, play crucial roles in habit formation, procedural learning, and skill acquisition. When you first learn to drive, you must consciously think about every action, checking mirrors, operating the pedals, turning the wheel. With practice, these actions become automatic, controlled by the basal ganglia rather than conscious deliberation. This transition from conscious to automatic processing frees up your limited attentional resources for other tasks, allowing you to drive while having a conversation or listening to music, demonstrating elegant efficiency of the brain's multiple learning and memory systems. Fact number 31 explores the role of emotion in memory. The amygdala, an almond-shaped structure deep in the temporal lobe involved in emotional processing, plays a critical role in modulating memory strength. Emotionally significant events, whether positive or negative, tend to be remembered more vividly and for longer than emotionally neutral experiences. This makes evolutionary sense. Remembering dangerous situations, threatening individuals, or sources of reward increases survival and reproductive success. The amygdala communicates with the hippocampus and cortical areas to enhance the consolidation of emotionally important memories. This is why you probably remember exactly where you were and what you were doing during major emotional events in your life. Falling in love, receiving shocking news, experiencing profound beauty, while forgetting countless mundane moments from those same time periods. Our 32nd fact reveals a counterintuitive finding about forgetting. Research published in Nature Review's Neuroscience suggests that forgetting isn't simply a failure of memory, but an active process that helps you function more effectively. The brain seems to deliberately forget less relevant information, allowing you to focus on what's most important for current goals and future decision-making. If you remembered every single detail of every experience with perfect clarity, you might struggle to extract general principles, recognize patterns, or apply past learning to new situations. Strategic forgetting declutters your mental workspace, prevents interference between similar memories, and helps you adapt to changing environments. What feels like a bug in the system might actually be a crucial feature, allowing flexible cognition in a complex, dynamic world. Fact number 33 brings us to working memory, that limited capacity system that holds information temporarily while you actively think about it. When you hear a phone number and try to remember it long enough to dial, you're using working memory. When you read a sentence and must hold the beginning in mind while processing the end to understand the whole meaning, that's working memory at work. Research suggests that working memory can hold about 4 to 7 items at once, though this varies between individuals and across different types of information. The prefrontal cortex plays a critical role in maintaining and manipulating information in working memory, keeping relevant representations active even in the absence of external stimulation. This capacity for internal representation, for holding information in mind independent of immediate sensory input, underlies much of higher cognition. Reasoning, problem-solving, planning, and creativity all depend on this ability to think about things that aren't physically present. Our 34th fact explores the phenomenon of memory reconsolidation. When you recall a memory, bringing it from storage back into conscious awareness, that memory enters a temporarily labile state where it becomes susceptible to modification before being stored again, a process called reconsolidation. During this window, the memory can be updated with new information, strengthened, weakened, or even potentially erased. Some researchers are exploring whether this presents therapeutic opportunities for treating conditions like PTSD, where traumatic memories cause ongoing distress. If a traumatic memory could be recalled and then modified during the reconsolidation window, perhaps by introducing competing emotional associations or by blocking the molecular mechanisms of reconsolidation, it might be possible to reduce the emotional impact of the memory without completely erasing the factual information it contains. Fact number 35 reveals the surprising architecture of long-term memory storage. Different aspects of a single memory distribute across different brain regions. When you remember visiting your grandmother's house as a child, the visual details might be stored in visual cortex areas, the smell of her cooking in olfactory-related regions, the emotional feeling of safety and love in the amygdala and related limbic structures, and the conceptual knowledge about who your grandmother was in temporal lobe semantic memory areas. Retrieving that complete memory means reactivating all these distributed components and binding them together into a unified, conscious experience. This distributed storage explains why brain damage can create specific memory deficits. Someone might lose memory for faces but not names, or for visual details but not emotional content, depending on which regions suffered damage. It also means that your memories are truly neural networks, patterns of activation spanning multiple brain areas rather than single units stored in single locations. Section 4. The chemistry of consciousness, neurotransmitters and mood. Fact number 36 takes us into the chemical world that underlies your mental states. More than 100 different neurotransmitters have been identified in the human nervous system, each playing specific roles in brain function. These chemical messengers, released by neurons and acting on receptors, translate electrical signals into chemical communication, allowing neurons to influence each other in complex ways. Some neurotransmitters excite their target neurons, making them more likely to fire. Others inhibit them, reducing the likelihood of firing. This balance between excitation and inhibition, mediated by dozens of different chemical systems, creates the dynamic patterns of brain activity underlying everything you think, feel, and do. Our 37th fact explores one of the most famous neurotransmitters. Dopamine, often called the reward molecule or feel-good hormone, plays crucial roles in motivation, pleasure, motor control, and decision-making. When you accomplish a goal, eat delicious food, or experience something enjoyable, dopamine levels surge in specific brain circuits, creating feelings of pleasure and satisfaction. But dopamine's role extends beyond just pleasure. It signals the difference between expected and actual rewards, helping you learn which behaviors lead to good outcomes. The dopamine system can go awry in various conditions. Too little dopamine in certain pathways contributes to the motor symptoms of Parkinson's disease, while dysregulation of dopamine signaling plays roles in addiction, schizophrenia, and ADHD. Understanding dopamine has revolutionized treatment for these conditions and illuminated fundamental principles of how brains learn and motivate behavior. Fact number 38 brings us to another crucial brain chemical. Serotonin, a neurotransmitter derived from the amino acid tryptophan, influences mood, sleep, appetite, sexual desire, body temperature, and many other physiological processes. Interestingly, about 95% of your body's serotonin resides not in your brain, but in your gut, where it helps regulate intestinal movements and other digestive functions. In the brain, serotonin helps regulate mood and emotional well-being, which is why many antidepressant medications work by increasing serotonin availability at synapses. The relationship between serotonin and mood is complex and not fully understood, but there's a strong correlation between disrupted serotonin signaling and conditions like depression, anxiety disorders, and obsessive-compulsive disorder. The brain's serotonin system originates from a small cluster of neurons in the brainstem called the raphe nuclei, which project throughout the brain to influence neural processing in virtually every region. Our 39th fact reveals the extraordinary scope of a single neurotransmitter system. Although your brain contains only about 400,000 dopaminergic neurons, neurons that produce and release dopamine, these relatively few cells exert enormous influence on brain function through their extensive projections. These dopamine neurons send their axons to virtually every part of the forebrain, creating 4 major dopamine pathways that regulate movement, motivation, reward, executive function, and hormonal responses. This widespread influence from such a small population of neurons demonstrates the brain's efficiency in achieving broad effects through strategic connectivity rather than sheer numbers. It also means that damage to or loss of these dopamine neurons, as occurs in Parkinson's disease, can have devastating and wide-ranging consequences despite affecting such a small percentage of total brain cells. Fact number 40 explores the neurotransmitter of action and attention. Acetylcholine, discovered in 1921 as the first identified neurotransmitter, plays crucial roles in muscle activation, attention, arousal, memory, and learning. In the peripheral nervous system, acetylcholine acts at the neuromuscular junction to trigger muscle contraction. When you decide to move your arm, motor neurons release acetylcholine onto your arm muscles, causing them to contract. In the brain, acetylcholine helps regulate sleep-wake cycles, with high levels promoting wakefulness and attention. The cholinergic system, originating from several clusters of neurons in the basal forebrain and brainstem, projects widely throughout the cortex and hippocampus. Degeneration of cholinergic neurons contributes to the cognitive decline seen in Alzheimer's disease, which is why many Alzheimer's medications work by preventing the breakdown of acetylcholine, maintaining higher levels to partially compensate for the loss of these neurons. Our 41st fact brings us to the brain's primary excitatory neurotransmitter. Glutamate, the most abundant neurotransmitter in the nervous system, is responsible for fast excitatory transmission throughout the brain and spinal cord.
Every time you think, learn, or remember something, glutamate is almost certainly involved. This neurotransmitter acts on several types of receptors, including the NMDA receptor, that plays crucial roles in synaptic plasticity and memory formation. The discovery of how NMDA receptors work helped explain long-term potentiation, the cellular basis of learning and memory. However, too much glutamate activity can be harmful. Excessive stimulation, called excitotoxicity, can damage or kill neurons, contributing to injury from strokes, head trauma, and some neurodegenerative diseases. The brain carefully regulates glutamate levels, maintaining a delicate balance between sufficient activation for normal function and excessive activation that causes harm. Fact number 42 explores the primary inhibitory neurotransmitter in the brain. GABA, short for gamma-aminobutyric acid, acts to reduce neural activity, preventing excessive excitation that could lead to seizures or anxiety. About 20 to 40% of all synapses in the brain use GABA as their neurotransmitter, making it nearly as ubiquitous as glutamate. Many anti-anxiety medications, including benzodiazepines like Valium and Xanax, work by enhancing GABA's effects on its receptors, increasing inhibitory signaling and calming neural activity. Alcohol also affects GABA receptors, contributing to its sedating and anxiety-reducing effects. The balance between glutamate-driven excitation and GABA-driven inhibition creates the dynamic stability necessary for normal brain function. Enough excitation for active processing and learning, enough inhibition to prevent runaway activation and maintain control. Our 43rd fact reveals the neurotransmitter of stress and alertness. Norepinephrine, also called noradrenaline, plays crucial roles in the brain's arousal and attention systems and in the body's fight or flight response. The brain's norepinephrine system originates primarily in a small cluster of neurons called the locus coeruleus, located in the brainstem. Despite containing only about 15,000 neurons on each side of the brain, the locus coeruleus projects to virtually every region of the brain and spinal cord, allowing rapid modulation of neural processing throughout the nervous system. When you perceive danger or face a challenging situation, norepinephrine levels increase, sharpening attention,
enhancing memory formation for important events and preparing your body for action. Dysregulation of norepinephrine signaling contributes to conditions including depression, ADHD, and PTSD, making it a target for various medications. Fact number 44 brings us to the discovery of endogenous opioids. Your brain produces its own pain-relieving and pleasure-inducing chemicals called endorphins, which bind to the same receptors as external opioid drugs like morphine and heroin. The word endorphin combines endogenous and morphine, literally meaning morphine produced within. These chemicals contribute to runner's high, the pain relief seen during severe injuries, the pleasure of eating chocolate, and the blissful feelings associated with certain social bonds. The brain's endogenous opioid system includes several different peptides, endorphins, enkephalins, and dynorphins, that act on multiple receptor types throughout the brain and body. Understanding this system has illuminated both the brain's natural pain control mechanisms and the neural basis of opioid addiction, as external opioid drugs hijack these natural systems, producing artificially intense activation that the brain wasn't designed to handle. Our 45th fact explores a fascinating connection between gut and brain. The same neurotransmitters that operate in your brain also function in your enteric nervous system, the extensive network of neurons lining your gastrointestinal tract, sometimes called the second brain. This gut nervous system contains approximately 500 million neurons, more than in your spinal cord, and uses many of the same neurotransmitters as your brain, including serotonin, dopamine, and acetylcholine. The gut and brain communicate constantly through the vagus nerve and through hormonal signals carried in the bloodstream. This bidirectional gut-brain axis influences mood, cognition, and behavior, helping explain why digestive problems often accompany mental health conditions, and why mental stress affects digestive function. The realization that the gut microbiome, the trillions of bacteria in your digestive system, can influence brain chemistry and behavior through this axis has opened new research avenues in neuroscience and psychiatry. Fact number 46 reveals the role of a neurotransmitter in maintaining wakefulness. Histamine, better known for its role in allergic reactions, also functions as a neurotransmitter in the brain. Histamine neurons, located in the hypothalamus, help promote wakefulness and alertness.
This is why antihistamine medications, designed to block histamine receptors to reduce allergy symptoms, often cause drowsiness as a side effect. By blocking histamine receptors in the brain, they inadvertently reduce the histamine signaling that helps keep you awake. The histamine system also influences feeding behavior, motivation, and learning, demonstrating once again how a single neurotransmitter system can affect multiple aspects of brain function and behavior. Our 47th fact brings us to the complex interactions between neurotransmitter systems. These chemical messengers don't work in isolation. They constantly influence each other in intricate networks of interaction. Dopamine and serotonin, for instance, can both stimulate and inhibit each other, depending on the brain region and receptor types involved. Norepinephrine can modulate the effects of glutamate. GABA and glutamate work together to maintain the balance between excitation and inhibition. This crosstalk between systems allows for sophisticated regulation of brain states,
and explains why many psychiatric medications, while primarily targeting one neurotransmitter system, ultimately affect multiple systems. It also means that understanding mental illness and treating it effectively requires appreciating this complex choreography of interacting chemical systems, rather than focusing on a single neurotransmitter in isolation. Fact number 48 explores the timescales of neurotransmitter action. While the fast neurotransmitters like glutamate and GABA act within milliseconds to rapidly excite or inhibit neurons, other neurotransmitters work more slowly, modulating neural activity over seconds, minutes, or even longer timescales. These neuromodulatory systems, dopamine, serotonin, norepinephrine, acetylcholine, and others, don't typically cause neurons to fire or not fire directly. Instead, they adjust the sensitivity, excitability, or responsiveness of neurons and neural circuits, tuning the brain's information processing to match current needs. When you're stressed, norepinephrine release modulates sensory processing to make you more alert to potential threats. When you're expecting a reward, dopamine shifts circuit dynamics to enhance learning. This combination of fast transmission and slower modulation gives the brain both the speed to respond rapidly and the flexibility to adjust its processing strategies based on context. Our 49th fact reveals a surprising aspect of neurotransmitter synthesis. Your brain must manufacture most of its neurotransmitters from precursor molecules, often amino acids from the diet. Dopamine and norepinephrine, for instance, are synthesized from the amino acid tyrosine, which comes from protein in food. Serotonin derives from tryptophan, another amino acid found in protein-rich foods. This means that, to some extent, your diet influences your brain's ability to produce neurotransmitters. While eating tyrosine-rich foods won't cure Parkinson's disease, or eating tryptophan-rich foods won't cure depression, maintaining adequate dietary intake of these precursor molecules supports normal neurotransmitter synthesis. Some people experiment with tryptophan or tyrosine supplements to potentially enhance mood or mental performance, though the evidence for effectiveness remains mixed, and the brain has complex regulatory systems that prevent simple dietary changes from dramatically altering neurotransmitter levels. Fact number 50 brings us to a remarkable feature of neurotransmitter diversity. Different neurons, even within the same brain region, can express different combinations of neurotransmitters and receptors, creating an astonishing variety of signaling options. A single neuron might release multiple neurotransmitters simultaneously or under different conditions. Receptors for a given neurotransmitter come in multiple subtypes. Serotonin alone has at least 14 different receptor subtypes, each with different effects on the receiving neuron. This molecular diversity allows for incredibly precise and flexible neural communication, with the potential for highly specific pharmacological interventions. Modern drug development increasingly targets specific receptor subtypes rather than broadly affecting all receptors for a given neurotransmitter, promising medications with greater effectiveness and fewer side effects by more precisely modulating the brain's chemical signaling. Section 5. Emotions and the limbic system, the feeling brain. Fact number 51 takes us deep into the emotional heart of the brain. The limbic system, a network of structures including the amygdala, hippocampus, hypothalamus, thalamus, and parts of the cortex, governs emotions, motivation, and emotional memories. The term limbic comes from the Latin word for border, referring to these structures' location bordering the brainstem and lying beneath the cerebral cortex.
This ancient system,
present in some form in all vertebrates, generates the instinctive behaviors and emotional responses necessary for survival. Fear that makes you flee from danger, anger that prepares you to fight threats, pleasure that draws you toward beneficial resources, disgust that protects you from contamination. While the limbic system concept has evolved, and some neuroscientists debate its precise boundaries, the core idea remains valid. These interconnected structures work together to create your emotional life and link emotions to behavior, memory, and bodily responses. Our 52nd fact explores the amygdala, that almond-shaped structure that has become almost synonymous with fear and emotion. You have two amygdalae, one in each hemisphere, nestled in the temporal lobes near the hippocampus. The amygdala isn't a single unit, but a complex of approximately 13 different nuclei, each with specialized functions and connections. While the amygdala is most famous for its role in fear responses, research shows it processes all emotionally significant information, whether positive or negative, pleasant or unpleasant. It helps you recognize emotions in other people's faces, attach emotional significance to memories, make rapid threat assessments, and generate appropriate emotional responses to situations. People with bilateral amygdala damage, a rare condition caused by diseases like Urbach-Vita disease, show impaired ability to recognize fear in facial expressions and reduced experience of fear themselves, though they can still experience other emotions. Fact number 53 reveals the amygdala's role in the speed of emotion. The amygdala receives sensory information through two pathways, a fast, direct route from the thalamus and a slower route through the cortex. The fast pathway, called the low road by neuroscientist Joseph Ledoux, allows the amygdala to begin processing potential threats before the cortex has fully analyzed the sensory information. This explains why you might jump back from a curved stick on a hiking trail before consciously recognizing it's not a snake, or why you might feel fear upon hearing a loud sound before knowing what caused it. The amygdala can trigger defensive responses through its connections to the hypothalamus and brainstem within milliseconds, a speed that could be life-saving in truly dangerous situations. The slower cortical pathway then provides detailed information, allowing you to correct false alarms. You realize it's a stick, not a snake, and your fear subsides. This two-pathway architecture prioritizes speed of response while still allowing for thoughtful evaluation, an elegant solution to the problem of responding adaptively in a potentially dangerous world. Our 54th fact explores the amygdala's contribution to emotional learning. Classical fear conditioning, one of the most studied forms of learning, depends critically on the amygdala. When a neutral stimulus, like a tone, is paired with an aversive event, like a mild shock, the amygdala learns this association rapidly, often in a single trial. Thereafter, the tone alone triggers fear responses, increased heart rate, stress hormone release, freezing behavior in animals, or heightened vigilance in humans. This ability to learn danger associations quickly has obvious survival value, allowing organisms to predict and avoid threats based on past experience. The cellular mechanisms of fear conditioning in the amygdala have been worked out in remarkable detail, involving the strengthening of synaptic connections between neurons encoding the tone and neurons responding to the shock, mediated by the same NMDA receptors that underlie memory formation in the hippocampus. Understanding amygdala-based fear learning has important therapeutic implications for treating anxiety disorders and PTSD. Fact number 55 reveals the complex role of the amygdala in social behavior. Beyond processing basic emotions like fear, the amygdala contributes to recognizing and interpreting social cues, judging trustworthiness, processing information about social hierarchies, and navigating complex social situations. Studies show that amygdala activity increases when people view faces with direct eye contact versus averted gaze, when evaluating trustworthy versus untrustworthy faces, and when considering social violations of norms. The amygdala is larger in people with larger social networks, suggesting a relationship between amygdala structure and social cognitive demands. Individuals with autism spectrum disorders, who often struggle with social cognition, show atypical patterns of amygdala structure and function. The amygdala doesn't operate alone in social cognition. It works closely with prefrontal cortex regions and other structures. But its role in processing the emotional and motivational aspects of social information appears crucial for normal social functioning.
Our 56th fact brings us to the prefrontal cortex and its interactions with the amygdala. The prefrontal cortex, the most forward part of the frontal lobes and the last brain region to fully mature, plays critical roles in executive function, decision-making, planning, and emotional regulation. It has strong reciprocal connections with the amygdala, allowing a constant conversation between these regions. The prefrontal cortex can modulate amygdala activity, providing top-down regulation of emotions. When you consciously try to calm yourself down during stress, when you reappraise a situation to feel less upset about it, when you suppress an emotional response to maintain composure, you're engaging prefrontal amygdala interactions. People with damage to certain prefrontal regions show poor emotional control and inappropriate social behavior despite intact amygdalae, while those with strong prefrontal function can often regulate emotions effectively even when experiencing high amygdala activation. Understanding this partnership between emotional generation in limbic structures and emotional regulation in prefrontal cortex has transformed approaches to treating mood and anxiety disorders. Fact number 57th explores the role of emotions in decision-making. The neuroscientist Antonio Damasio, studying patients with damage to the ventromedial prefrontal cortex, a region involved in processing emotions and their relation to decisions, found something surprising. These patients maintained normal intelligence and reasoning abilities, but made catastrophically bad life decisions, particularly in social and financial domains. Damasio proposed that emotions, rather than being the enemy of rationality, actually provide essential information for good decision-making. Emotions encode value, marking options as desirable or undesirable based on past experience and current goals. Without emotional input, decision-making becomes paralyzed by endless deliberation or guided only by explicit logical analysis that misses crucial subjective factors. This somatic marker hypothesis, as Damasio called it, suggests that gut feelings generated by limbic structures provide necessary guidance for navigating complex decisions, particularly those involving uncertainty, social factors, or long-term consequences.
Our 58th fact reveals the phenomenon of emotional contagion. Emotions can spread from person to person through automatic mimicry and physiological synchronization, a process involving multiple brain systems, including the amygdala and mirror neuron systems. When you see someone smile, microactivation of your own facial muscles occurs, even if you don't consciously smile back. And this facial feedback can subtly influence your own emotional state. When you're in a room with someone experiencing strong emotion, your own nervous system begins to echo their physiological state. Your heart rate might synchronize. Your posture might mirror theirs. Your amygdala might activate in response to their emotional expressions. This automatic emotional resonance served important social functions throughout human evolution, facilitating empathy, coordinating group responses to threats or opportunities, and strengthening social bonds. It operates largely below conscious awareness, a testament to how deeply social your brain truly is. Fact number 59 explores the relationship between emotions and memory consolidation. Not only does the amygdala enhance initial memory encoding of emotional events, it also influences how those memories consolidate during sleep and persist over time. When you experience something emotionally significant, the amygdala communicates with the hippocampus and neocortex to prioritize that memory for consolidation. During subsequent sleep, particularly REM sleep, emotional memories show enhanced replay and strengthening. This explains why emotional events, first kiss, traumatic accidents, moments of triumph or loss, remain vivid decades later, while mundane events from the same periods fade. The amygdala essentially flags certain memories as important enough to preserve, directing the limited resources of the consolidation system toward experiences that matter. This makes sense from an evolutionary perspective. Remembering emotionally significant events provides information crucial for future survival and reproduction. Our 60th fact reveals a fascinating aspect of emotional asymmetry in the brain. Research suggests that the left and right amygdalae have somewhat different functions. The right amygdala appears more involved in rapid, automatic emotional responses and in processing negative emotions, while the left amygdala contributes more to sustained emotional processing and positive emotions. Studies show that electrical stimulation of the right amygdala more reliably produces negative emotions like fear and sadness, while left amygdala stimulation can produce either pleasant or unpleasant emotions.
This hemispheric specialization for emotion extends beyond the amygdala. The right hemisphere appears generally more involved in emotional processing than the left, while the left hemisphere shows greater involvement in language and analytical thinking. However, these lateralization patterns represent tendencies rather than absolute divisions, and both hemispheres contribute to virtually all mental functions. Fact number 61 brings us to the hypothalamus, a small structure weighing only about 4 grams, yet wielding enormous influence over bodily functions and emotions. The hypothalamus sits just below the thalamus and above the brainstem, forming part of the limbic system. Despite its tiny size, it controls the autonomic nervous system, regulates body temperature, hunger, thirst, sleep-wake cycles, and circadian rhythms, coordinates stress responses, and controls the pituitary gland, thereby influencing the entire endocrine system. When you feel hungry, thirsty, sleepy, or sexually aroused, your hypothalamus is generating these drives.
When you face stress, the hypothalamus initiates the cascade of hormonal responses, preparing your body for action. This tiny region integrates information about your internal state with information about the external environment and emotional significance, orchestrating physiological and behavioral responses appropriate to the current situation. Our 60-second fact explores the fascinating phenomenon of stress and the limbic system. When you encounter a stressor, the amygdala and hypothalamus work together to activate two major stress response systems. The fast response involves the sympathetic nervous system, with the hypothalamus sending signals that cause the adrenal glands to release epinephrine and norepinephrine within seconds, creating the immediate fight-or-flight response: rapid heartbeat, quickened breathing, heightened alertness. The slower response involves the hypothalamic pituitary adrenal axis, culminating in release of cortisol over minutes to hours, which helps mobilize energy stores and modulate immune and inflammatory responses. These stress systems evolved to help organisms respond to acute threats, fleeing predators, fighting for resources. But chronic activation in response to modern psychological stressors can harm health, affecting cardiovascular function, immune function, and even brain structure, particularly in the hippocampus, which contains many receptors for stress hormones and can shrink under prolonged stress exposure. Fact number 63 reveals the role of the anterior cingulate cortex in emotional experience. This strip of cortex running along the midline of the brain, forming part of the limbic system, appears crucial for the conscious experience of emotion. Neuroimaging studies show that anterior cingulate activity correlates with reported emotional intensity across different emotions. The anterior cingulate has extensive connections with both the amygdala and prefrontal cortex, positioning it to integrate automatic emotional reactions with conscious appraisal and regulation. It also plays roles in error detection, conflict monitoring, and decision-making, particularly decisions involving uncertain outcomes or emotional factors. Lesions to the anterior cingulate can produce emotional blunting, where patients report difficulty feeling emotions even though they may still show some emotional behaviors, suggesting this region contributes specifically to the subjective, felt quality of emotional experience. Our 64th fact explores a remarkable treatment that reveals the specificity of emotional brain circuits. Deep brain stimulation, where electrodes implanted in specific brain regions deliver carefully controlled electrical stimulation, can dramatically affect emotional states depending on the target location. Stimulation of certain sites within the limbic system can trigger intense feelings of happiness, sadness, fear, or anger, demonstrating that these emotional states correspond to activity in specific neural circuits. Some patients receiving deep brain stimulation for treatment-resistant depression report immediate mood improvement when the stimulator is turned on and return of depressive symptoms when it's turned off, suggesting that stimulation is artificially activating or inhibiting circuits involved in mood regulation. While still an experimental treatment for psychiatric conditions, deep brain stimulation has proven effective for some patients and has provided valuable insights into the neural circuitry underlying emotional disorders. Fact number 65 brings us to the insula, a region of cortex folded deep within the lateral sulcus of each hemisphere, which appears crucial for emotional awareness and interoception, the sense of your body's internal state. The insula receives information about bodily states, heartbeat, breathing, gut sensations, pain, temperature, and integrates this with emotional and cognitive information. It activates when you feel disgusted, when you experience pain, when you crave drugs during addiction, when you feel empathy for others' suffering, and when you become aware of your own emotional states. The anterior insula, in particular, seems important for conscious emotional experience, perhaps by representing the bodily changes associated with emotions. This region's role in emotional awareness suggests that emotions aren't purely mental phenomena, but intimately involve representations of bodily states, supporting theories that view emotions as patterns of bodily response that become conscious through brain mechanisms that monitor those responses. Section 6. Sleep, Dreams, and the Resting Brain. Fact number 66 takes us into the mysterious world of sleep. Humans spend approximately 1/3 of their lives sleeping. And while we're still unraveling exactly why this is necessary, we know it's essential for survival. Rats deprived of sleep die within 2 to 3 weeks, faster than they would die from starvation. Humans deprived of sleep experience progressive cognitive impairment, emotional instability, hallucinations, and, if deprivation continues long enough, death. Sleep appears to serve multiple critical functions: consolidating memories, clearing metabolic waste from the brain, restoring cellular energy, regulating immune function, processing emotions, and allowing the brain to reorganize and prepare for waking activity. The fact that evolution preserved sleep despite its apparent disadvantages, making organisms vulnerable to predators, reducing time for gathering food and reproducing, suggests it must provide benefits important enough to outweigh these costs. Our 67th fact explores the structure of sleep. Sleep isn't a uniform state, but consists of distinct stages that cycle throughout the night. Non-REM sleep includes 3 stages ranging from light to deep sleep, characterized by progressively slower brain waves. The deepest stage, sometimes called slow wave sleep because of the large, slow delta waves dominating the EEG during this phase, is when growth hormone releases, tissue repair occurs, and immune function strengthens. REM sleep, named for the rapid eye movements that occur during this stage, is when most vivid dreaming happens. Brain activity during REM sleep resembles waking activity in many ways, hence its older name, paradoxical sleep. Yet the body experiences temporary paralysis, preventing you from acting out dreams. A complete sleep cycle through all stages takes about 90 to 110 minutes, and you typically experience 4 to 6 cycles per night. Fact number 68 reveals the role of sleep in memory. While you sleep, your brain actively processes and consolidates memories from the day. During slow-wave sleep, the hippocampus replays patterns of activity from recent experiences, gradually transferring memories to the neocortex for long-term storage. Studies show that people allowed to sleep after learning perform significantly better on memory tests than those kept awake, even when tested days or weeks later. Specific memories can be strengthened during sleep through targeted memory reactivation. If people learn associations between information and particular cues, then presenting those cues during subsequent sleep enhances memory for the associated information. REM sleep appears particularly important for consolidating procedural memories and integrating new information with existing knowledge. The sleeping brain isn't resting passively, but actively working to organize, strengthen, and integrate the day's experiences. Our 69th fact brings us to the brain chemicals regulating sleep and wakefulness. Multiple neurotransmitter systems interact to control sleep-wake states. Orexin neurons in the hypothalamus promote wakefulness. When these neurons malfunction, narcolepsy results, causing uncontrollable sleep attacks. Adenosine accumulates in the brain during waking hours, increasing sleep pressure. Caffeine promotes alertness by blocking adenosine receptors. Melatonin, produced by the pineal gland in response to darkness, promotes sleepiness and helps regulate circadian rhythms. The neurotransmitters norepinephrine, serotonin, and histamine promote wakefulness, while GABA promotes sleep. This complex interplay between promoting wakefulness and promoting sleep creates stable states. When you're awake, wakefulness-promoting systems actively inhibit sleep-promoting systems, and vice versa, with relatively rapid transitions between states. Understanding these systems has led to development of medications that promote sleep or wakefulness by targeting specific components of this regulatory network.
Fact number 70 explores the phenomenon of dreaming. People dream during all stages of sleep, though REM dreams tend to be more vivid, bizarre, and narrative than non-REM dreams, which are often more thought-like and mundane. On average, people have 4 to 7 dreams per night, though most are forgotten shortly after waking. Dreams have fascinated humans throughout history, inspiring religious and mystical interpretations, psychological theories, and scientific investigation. Recent research challenges some common assumptions about dreaming. While REM sleep and dreaming are associated, they're controlled by different brain mechanisms. You can have REM sleep without dreaming or dreams without REM sleep, though they usually co-occur. Dreaming requires an intact forebrain, particularly regions involved in imagery and narrative, but doesn't require the brainstem mechanisms that generate REM sleep. Our 71st fact reveals what happens in the dreaming brain. During REM sleep and vivid dreaming, brain imaging shows that visual and motor cortex areas activate intensely, consistent with the visual and action-filled content of dreams. The limbic system, including the amygdala, shows high activity, perhaps explaining dreams' often emotional quality. Interestingly, the dorsolateral prefrontal cortex, involved in logical reasoning and executive control, shows reduced activity during REM sleep, which might explain why dreams often violate logic, jump between scenes without coherent transitions, and why you rarely realize you're dreaming while it's happening, that metacognitive awareness requires prefrontal function that's offline during REM sleep. The primary sensory and motor areas of the brain become disconnected from the outside world during REM sleep, which is why external stimuli are largely blocked out, and why motor commands generated during dreams don't result in actual movement. Fact number 72 brings us to the role of dreams in emotional processing. Some theories propose that dreaming, particularly REM dreaming, helps process emotions and stress. During REM sleep, the brain appears to reprocess emotional experiences from the day, but in a neurochemical environment where stress hormones like norepinephrine are absent. This might allow emotional memories to be integrated and stored without the intense emotional charge they initially carried, a kind of overnight therapy. People with REM sleep disorders or who are REM deprived show increased emotional reactivity and difficulty regulating emotions. PTSD often includes nightmares that replay traumatic experiences, perhaps representing a failure of this normal emotional processing function. Some evidence suggests that REM sleep and dreaming help make distant or creative associations between ideas, potentially contributing to insight and problem-solving, the origin of the advice to sleep on it when facing difficult decisions.
Our 73rd fact explores the content of dreams. Research by analyzing thousands of dream reports reveals consistent patterns. Dreams typically feature the dreamer as a character interacting with other people in familiar or realistic settings, though strange elements commonly intrude. Visual imagery dominates, though other sensory modalities appear less frequently. Emotions in dreams are common, with anxiety and fear appearing more often than positive emotions. Recent experiences incorporate into dreams, but usually not in direct replay. Instead, they blend with older memories and fictional elements in ways that seem meaningful to the dreamer, yet bizarre upon waking reflection. Some common dream themes appear across cultures: being chased, falling, appearing naked in public, being unprepared for an exam, suggesting either universal psychological concerns or common physiological experiences during sleep that the brain interprets through these narrative frameworks. Fact number 74 reveals a fascinating finding about dream content and learning. When people learn a complex task before sleep, dreams about that task predict improved performance after waking. In one study, participants learning to navigate a virtual maze showed better post-sleep performance if they dreamed about the maze compared to those who learned the maze but didn't dream about it. Even fragmentary dream imagery related to the learning task correlated with improvement. This suggests that dreams might reflect memory consolidation processes occurring during sleep. The dream content provides a window into the brain's work of integrating new learning with existing knowledge. The relationship isn't that dreaming causes improvement, but that both the dream content and the performance improvement result from the brain actively processing the learned material during sleep.
Our 75th fact brings us to lucid dreaming, where the dreamer becomes aware they're dreaming while the dream continues. About half of people report experiencing at least one lucid dream, though frequent lucid dreamers are rare. During lucid dreams, dreamers can sometimes exert control over dream content, directing the narrative or choosing actions within the dream. Brain imaging of lucid dreamers shows increased activation of prefrontal cortex regions compared to normal dreaming, particularly areas involved in self-awareness and metacognition, the same regions that show reduced activity during typical REM dreams. Some people practice techniques to increase lucid dreaming frequency, and researchers are exploring potential therapeutic applications, such as using lucid dreaming to rehearse behaviors, overcome nightmares, or process difficult emotions in a safe environment where the dreamer knows the experiences aren't real. Fact number 76 explores what happens during deep, slow-wave sleep. This stage, characterized by high-amplitude, low-frequency delta waves on EEG, appears crucial for physical restoration and memory consolidation. During slow-wave sleep, growth hormone releases, promoting tissue growth and repair. The immune system strengthens with increased production of certain immune cells and inflammatory mediators that help fight infection. Metabolic rate slows, body temperature drops, and cardiovascular stress decreases, giving these systems time to recover from daily demands. The brain's glymphatic system, a waste clearance system discovered in 2012, becomes most active during sleep, flushing out metabolic waste products, including beta-amyloid proteins that accumulate during waking and, if not cleared, can contribute to Alzheimer's disease. Sleep deprivation reduces slow-wave sleep and impairs all these restorative processes, helping explain why chronic sleep loss harms health in so many ways. Our 77th fact
reveals how the brain regulates when you sleep. The circadian rhythm, your internal biological clock, runs on a cycle of approximately 24 hours, governing sleep-wake patterns, body temperature, hormone release, and other physiological processes. This clock resides primarily in the suprachiasmatic nucleus, a small region of the hypothalamus containing about 20,000 neurons. These neurons generate rhythmic patterns of activity through interlocking feedback loops of gene expression, creating an autonomous oscillation with a period close to, but not exactly, 24 hours. Light hitting photoreceptive cells in the retina sends signals to the suprachiasmatic nucleus, synchronizing this internal clock to the external day-night cycle. Without light input, as experienced by people living in constant darkness or by blind individuals, the circadian rhythm continues but often drifts from 24 hours, leading to the internal clock becoming desynchronized with the environmental cycle, which can cause sleep disorders. Fact number 78 brings us to individual differences in sleep needs and timing. While adults generally need 7 to 9 hours of sleep per night, individuals vary considerably. A few rare individuals carry genetic variants, allowing them to function well on significantly less sleep, sometimes as little as 4 to 6 hours per night, though such short sleepers represent a tiny fraction of the population. Most people claiming they need minimal sleep are actually sleep-deprived and suffering health consequences. People also differ in their chronotype, whether they're morning larks, who naturally wake early and feel most alert in morning, or night owls, who prefer staying up late and sleeping late. These differences have genetic components and change across the lifespan, with teenagers tending strongly toward night owl patterns, perhaps explaining why forcing teenagers to attend school early in the morning works against their biological rhythms and impairs learning. Our 79th fact explores a curious sleep phenomenon. Sleep paralysis, where you wake up unable to move for several seconds or minutes, occurs when the brain emerges from REM sleep before ending the muscle paralysis that normally accompanies REM. During these episodes, you're conscious and your eyes can move, but other muscles remain paralyzed. This can be terrifying, especially if accompanied by hallucinations, visual imagery, sensations of pressure on the chest, or feelings of presence in the room, which occur because parts of the dreaming brain remain active. Sleep paralysis happens to about 8% of people at least once, more commonly during sleep deprivation, irregular sleep schedules, or sleeping on one's back. While frightening, it's harmless and typically resolves within minutes. Understanding the neurological basis of sleep paralysis demystifies these experiences and reduces the anxiety that can make them worse.
Fact number 80 reveals what happens when you don't get enough sleep. Even partial sleep deprivation, like getting 6 hours per night instead of 8, accumulates sleep debt that impairs cognitive function, mood, and health. After staying awake for 24 hours, cognitive impairment resembles that of legal intoxication. Chronic sleep deprivation increases risk for obesity, diabetes, cardiovascular disease, depression, anxiety, and Alzheimer's disease. It weakens immune function, making you more susceptible to infections. Sleep deprivation impairs the prefrontal cortex particularly, reducing impulse control, decision-making ability, and emotional regulation, while the amygdala becomes hyperactive, leading to increased emotional reactivity. Memory consolidation suffers when you don't sleep enough. Even a single night of poor sleep affects next-day functioning. and chronic sleep restriction creates a spiral where impaired judgment prevents recognizing how impaired you've become. Prioritizing adequate sleep represents one of the most powerful steps you can take for brain health and general well-being. The human nervous system begins forming just 3 weeks after conception, when a sheet of cells in the developing embryo folds to form the neural tube, which will become the brain and spinal cord. By 4 weeks, major brain regions can be recognized in primitive form. By 6 months of gestation, the characteristically wrinkled cerebral cortex becomes visible. During peak periods of prenatal development, the brain produces approximately 250,000 neurons per minute, an astonishing rate of neurogenesis that creates the billions of neurons that will form the mature brain. Most of these neurons are produced before birth, though the brain continues growing rapidly after birth through proliferation of glial cells, extension of neural processes, formation of new synapses, and myelination of nerve fibers. Our 80-second fact explores the explosion of connectivity in early life. A newborn baby's brain contains most of the neurons it will ever have, but it has relatively few synaptic connections between them. In the first few years of life, the brain undergoes extraordinary synaptic proliferation, forming connections at an incredible rate. By age 2 or 3, a child's brain contains about twice as many synapses as an adult brain, more connections than will ultimately be needed. This overproduction of synapses provides a substrate for experience to sculpt brain connectivity. Synapses that are frequently activated strengthen and stabilize, while inactive synapses gradually weaken and disappear through a process called synaptic pruning. This experience-dependent refinement of connectivity allows the developing brain to adapt to its specific environment, optimizing neural circuits based on the particular sensory, motor, cognitive, and social demands the child faces. Fact number 83 reveals critical periods in development. During certain windows of time, the brain shows heightened plasticity for acquiring particular skills or abilities. The visual system, for instance, has a critical period in early childhood when normal visual experience is essential for proper development. Children who have cataracts blocking visual input during this period develop permanent visual deficits, even if the cataracts are later removed, because the visual cortex failed to develop properly without normal stimulation. Language acquisition shows a similar sensitive period, with children effortlessly acquiring their native language or languages during early childhood, but adults struggling much more with second language learning. These critical periods result from complex developmental programs involving changes in neurotransmitter systems, myelination, expression of genes regulating plasticity, and maturation of inhibitory circuits. While the brain retains significant plasticity throughout life, these early periods of heightened plasticity provide special opportunities for learning that, if missed, may be difficult or impossible to recapture. Our 84th fact brings us to the continued development of the adolescent brain. Although the brain reaches about 90% of its adult size by age 6, development continues well into the 20s. The prefrontal cortex, responsible for planning, impulse control, decision-making, and reasoning, is one of the last brain regions to fully mature. Myelination of prefrontal pathways continues into the mid-20s, improving the efficiency and integration of prefrontal function with other brain regions. This protracted development helps explain characteristic features of adolescent behavior: increased risk-taking, greater sensitivity to peer influence, heightened emotional reactivity, and sometimes impulsive decision-making. The limbic system, including reward-related structures, develops faster than the prefrontal control system, creating a temporary imbalance between emotional drives and regulatory control. Understanding this neurodevelopmental trajectory has implications for education, policy, and how society views adolescent decision-making and responsibility. Fact number 85 explores sex differences in brain development timing. While males generally have slightly larger brains than females on average, this size difference doesn't correlate with intelligence. Brain structure and connectivity patterns matter more than raw size. Girls' brains tend to develop slightly faster than boys' brains, particularly in regions involved in language and fine motor control, which might contribute to girls' earlier acquisition of certain skills. Within the amygdala, development follows different time courses in males and females, with male amygdala volumes peaking later. These developmental differences, combined with hormonal influences during puberty, might contribute to sex differences in the prevalence and timing of certain psychiatric conditions. Autism and ADHD, for instance, are more commonly diagnosed in males and typically appear in childhood, while depression and anxiety disorders become more prevalent in females after puberty. However, individual variation within each sex far exceeds average differences between sexes, and sociocultural factors profoundly influence development, making simple biological explanations of sex differences inadequate. Our 86th fact reveals what happens to the brain as we age. Starting around age 30, the brain gradually begins to shrink, losing about 5% of its weight per decade. The prefrontal cortex and hippocampus show particularly prominent age-related shrinkage, which might contribute to age-related declines in memory and executive function. The number of neurons declines with age. The hippocampus stimulates release of growth factors that support neural health, reduces inflammation, and improves vascular health. Cognitive engagement, continuing to learn new things, solving puzzles, reading, maintaining social connections, helps build cognitive reserve, the brain's resilience to age-related changes. People with higher education and more mentally demanding careers show better preservation of cognitive function in old age, though whether education causes better outcomes or whether people with naturally higher cognitive abilities pursue more education remains debated. Managing cardiovascular risk factors, controlling blood pressure, cholesterol, and blood sugar, protects brain health since the brain depends critically on adequate blood supply, adequate sleep, stress management, maintaining social connections, and a healthy diet all contribute to cognitive health across the lifespan. Our 88th fact explores the concept of cognitive reserve. Two people with the same degree of age-related brain changes or even early Alzheimer's pathology can show vastly different levels of cognitive impairment. Some people develop dementia with relatively modest brain changes, while others maintain normal cognition despite considerable pathology. This variability relates to cognitive reserve, the brain's ability to maintain function despite damage through more efficient use of neural networks or recruitment of alternative strategies. Education, occupational complexity, multilingualism, musical training, and other forms of cognitive enrichment appear to build cognitive reserve, perhaps by creating more elaborate neural networks with greater redundancy, allowing the brain to compensate for focal damage by routing processing through alternative pathways. Building cognitive reserve throughout life may provide protection against age-related cognitive decline and delay the clinical manifestation of dementia. Fact number 89 reveals a hopeful finding about neurogenesis and aging. Although the rate of adult neurogenesis declines with age, new neurons continue to be born in the hippocampus, even in elderly adults. Studies examining post-mortem brain tissue from people in their 70s and 80s found neural precursor cells and newly generated neurons, challenging earlier beliefs that adult neurogenesis stopped earlier in life. Exercise, learning, and environmental enrichment can enhance neurogenesis even in aged brains, suggesting that lifestyle factors influence brain plasticity throughout life. While neurogenesis represents only one component of brain health and plasticity, its continuation into old age challenges pessimistic views that the aging brain is inevitably rigid and declining, instead supporting a more nuanced understanding where age brings both vulnerabilities and retained capacities for adaptation. Our 90th fact brings us to the mysteries of consciousness across development. When does consciousness begin? Newborns clearly experience sensations and emotions, but their subjective experience differs vastly from adult consciousness. The development of self-awareness, the ability to recognize oneself in a mirror, typically emerges around 18 months. The capacity for episodic memory, mentally traveling back in time to re-experience past events, develops later, around age 3 to 4, which might relate to childhood amnesia and why you don't remember your early years. The gradual development of language transforms the nature of conscious experience, allowing for inner speech, narrative thought, and the ability to reflect on one's own mental states. Understanding consciousness development remains one of the great challenges in neuroscience, requiring us to grapple with the relationship between neural complexity, information processing, and subjective experience, questions that push the boundaries of scientific inquiry into philosophical territory. The retina,
The light-sensitive tissue at the back of your eye contains approximately 100 million photoreceptors that convert light into neural signals. These signals travel through the optic nerve to the brain, with each optic nerve containing about 1 million axons. Visual information first reaches the lateral geniculate nucleus of the thalamus, then projects to the primary visual cortex at the back of the brain. From there, information flows through two major pathways: the ventral stream, processing object identity, color and form, and the dorsal stream, processing spatial location, motion, and visual guidance of action. Processing vision requires approximately 30% of the cortex, more than any other sensory modality, highlighting vision's dominance in human perception. The speed of visual processing is remarkable. You can identify objects in photographs presented for just 13 milliseconds, demonstrating the brain's extraordinary ability to rapidly extract meaning from complex visual scenes.
Our 90-second fact explores the brain's construction of visual experience. What you see isn't a simple recording of the light entering your eyes, but an active construction by your brain. The blind spot, where the optic nerve exits the retina and there are no photoreceptors, creates a gap in your visual field that you never consciously perceive because your brain fills it in based on surrounding information. Your eyes make rapid movements called saccades several times per second, yet you experience stable vision rather than a jumpy image because the brain suppresses visual input during saccades and seamlessly integrates information across fixations. Color vision results from brain computations comparing signals from three types of cone photoreceptors. The specific wavelengths in the physical stimulus don't directly determine perceived color, as demonstrated by color constancy, where an object's perceived color remains stable despite changes in illumination that dramatically alter the wavelengths reflected from it. Vision is an inferential process, with the brain making best guesses about the external world based on ambiguous sensory input, priors based on past experience, and assumptions about how the physical world works. Fact number 93 reveals fascinating aspects of auditory processing. Your auditory system can detect incredibly subtle differences in sound timing, localizing sound sources based on microsecond differences in when sound reaches each ear. This exquisite temporal precision requires specialized neurons in the brainstem that can respond to sub-millisecond timing differences, making the auditory system one of the most temporally precise systems in the brain. The cochlea, the snail-shaped structure in the inner ear where sound waves get converted to neural signals, contains about 16,000 hair cells organized along its length, with different locations responding to different frequencies. This spatial organization of frequency is maintained throughout the auditory pathway, creating tonotopic maps in multiple brain areas. The primary auditory cortex, located on the superior temporal gyrus, receives processed auditory information and extracts complex features including pitch, timbre, spatial location, and temporal patterns. Beyond this primary area, specialized regions process different aspects of sound, including areas specifically responsive to speech sounds, others to music, and yet others to environmental sounds. Our 94th fact brings us to the chemical senses, taste and smell. Your sense of smell is mediated by approximately 400 different types of olfactory receptors in the nose, each responsive to different molecular features. Combinations of activated receptors allow you to distinguish thousands of different odors. Olfactory information projects directly to the olfactory cortex and amygdala without passing through the thalamus first, unlike other senses, creating the tight connection between smells and emotions and memories. Why a particular scent can instantly transport you to a childhood memory or evoke powerful feelings. Taste, mediated by taste receptor cells in taste buds on the tongue and elsewhere in the mouth, detects 5 basic qualities: sweet, sour, salty, bitter, and umami, though what we commonly call taste actually involves substantial contribution from smell. This is why food tastes bland when you have a cold blocking your sense of smell. The taste receptors are working fine, but you're missing the rich olfactory information that normally combines with taste to create flavor perception. Fact number 95 explores the somatosensory system processing touch, temperature, pain, and proprioception, your sense of body position. Touch receptors in your skin come in multiple types, each specialized for detecting different aspects of tactile stimulation. Some respond to light touch, others to pressure, vibration, or stretch. The density of these receptors varies across the body, with fingertips having extremely high receptor density, allowing fine tactile discrimination, while the back has much lower density. This distribution is reflected in the somatosensory cortex, where body parts with greater sensitivity receive disproportionately large cortical representation, creating the famous sensory homunculus, a distorted map of the body with huge hands and lips relative to the trunk. Pain processing is complex, involving multiple pathways and brain areas. Acute pain activates somatosensory cortex, insula, and anterior cingulate cortex, among other regions. Chronic pain can cause changes in brain structure and function, with some evidence that the pain itself contributes to these changes, creating a vicious cycle where chronic pain alters brain circuits in ways that maintain or amplify pain perception. Our 96th fact reveals the multisensory nature of perception. While we often think of senses as separate channels, the brain integrates information across modalities to create unified perceptual experiences. The ventriloquism effect, where you perceive speech as coming from a puppet's moving mouth rather than the ventriloquist's stationary mouth, demonstrates vision's influence on auditory localization. The McGurk effect shows auditory speech perception influenced by visual lip movements. When you hear ba while watching someone mouth ga, you might perceive da, a sound that wasn't present in either the auditory or visual input alone. Taste perception is influenced by smell, color, texture, and even by sounds. Studies show that listening to high-pitched sounds makes food taste sweeter, while low-pitched sounds enhance bitterness. The brain contains multisensory convergence zones where information from different senses combines, and this integration begins earlier in sensory processing than once thought, challenging the traditional view of separate sensory streams that only converge at higher levels. Fact number 97 brings us to attention and consciousness. Your brain receives far more sensory information than you can consciously process, so attention acts as a filter, selecting certain information for detailed processing while suppressing the rest. This selective attention can be remarkably powerful. In the famous invisible gorilla experiment, people watching a video and counting basketball passes often completely fail to notice a person in a gorilla suit walking through the scene, demonstrating inattentional blindness. Attention operates through multiple mechanisms, modulating the gain of sensory neurons, biasing competition between stimuli for neural representation, synchronizing neural oscillations in relevant brain areas, and engaging prefrontal and parietal control networks that coordinate attentional allocation based on current goals. The neural systems supporting attention overlap substantially with those supporting consciousness, leading some researchers to propose that attention and consciousness are deeply intertwined, with attention being necessary for full conscious awareness of stimuli. Our 98th fact explores the nature of consciousness itself, perhaps the deepest mystery in neuroscience. How does the physical activity of neurons give rise to subjective experience, the qualitative feeling of seeing red, hearing music, feeling pain, or experiencing any sensation? This hard problem of consciousness, as philosopher David Chalmers called it, remains unsolved. Scientists have identified neural correlates of consciousness, particular patterns of brain activity that reliably accompany conscious experiences, but identifying correlation doesn't explain how or why neural activity produces subjective experience. Some theories propose that consciousness emerges from integrated information across brain networks. Others suggest it relates to specific types of neural oscillations or particular patterns of feedback between brain areas. The prefrontal and parietal cortex appear crucial for consciousness, as damage to these areas can eliminate conscious perception while leaving unconscious processing intact. Consciousness likely exists on a continuum rather than being all or nothing, with different levels of awareness in different states: waking, dreaming, anesthesia, vegetative states, each associated with different patterns of brain connectivity and information flow. Fact number 99 reveals the brain's remarkable ability to adapt to sensory loss or change. When people lose one sense, their remaining senses often sharpen, and brain areas normally devoted to the lost sense get recruited for other functions, a beautiful example of cross-modal plasticity. Blind individuals show enhanced auditory and tactile abilities, and their visual cortex, deprived of its usual input, responds to sounds and touch. Deaf individuals have enhanced peripheral vision and superior abilities to detect visual motion. In a dramatic demonstration of brain plasticity, people can learn to use novel sensory substitution devices that translate visual information into auditory or tactile patterns. After training,
Congenitally blind users of these devices activate their visual cortex when using the device, and they report perceptual experiences that feel visual despite the information arriving through different sensory channels. These adaptations demonstrate that brain regions aren't rigidly locked into specific functions, but can flexibly respond to whatever information they receive, with the specific computations they perform being more important than the particular sensory modality they typically process. Our 100th and final fact brings us full circle to consider the brain's fundamental nature. The brain is simultaneously biological tissue, information processor, generator of consciousness, and product of evolution. It's the source of everything that makes you uniquely you. Your memories, personality, skills, beliefs, emotions, and experiences all arise from this 3-pound mass of cells. The fact that you can read these words, understand their meaning, form mental images, feel emotions, and reflect on your own existence all depends on the properly coordinated activity of billions of neurons following the laws of physics and chemistry. Yet from this physical substrate emerges something that feels transcendent: consciousness, creativity, love, wonder, the capacity to contemplate the universe and our place in it. Your brain contains approximately 86 billion neurons, each connected to thousands of others through trillions of synapses, creating a network of staggering complexity. But even facts about your remarkable brain settle gently in your mind like snowflakes falling on still water. Consider the vast neural networks firing softly as you rest, pulses of electricity traveling along billions of axons at this very moment, chemical signals flowing across trillions of synapses, the endless conversation between neurons that never truly stops. Even now, as consciousness fades and sleep approaches, your brain continues its essential work, the synaptic connections being strengthened and pruned even now, refined by today's experiences, shaped by what you learned, what you felt, what mattered to you. The memories consolidating, moving from temporary hippocampal storage into the permanent archives of your cortex, being organized, indexed, connected to related knowledge. The waste products being cleared by that remarkable glymphatic system, flushing out the metabolic debris that accumulated during waking hours, proteins and toxins washed away in cleansing fluid flows. The restoration occurring throughout this extraordinary organ, damaged molecules being repaired, depleted resources being replenished, cellular machinery being maintained and renewed. Your brain never truly rests, even in the deepest sleep, even when you've completely lost conscious awareness of the world around you, even when you're lost in dreams or suspended in that void of deep, slow-wave sleep. Your brain continues its essential work, maintaining the miracle of your existence, keeping your heart beating, your lungs breathing, your body temperature regulated, your hormone levels balanced, all the 1,000 automatic processes that keep you alive proceeding without any conscious effort on your part. Tomorrow when you wake, your brain will have been subtly changed by today's experiences, by tonight's sleep, by the dreams you'll dream, by these very words you've heard flowing through your auditory system and lighting up comprehension networks across your temporal and frontal lobes. That's the beauty of neuroplasticity. You're never finished, never static, never permanently fixed in one configuration. You're always capable of learning, growing, adapting, changing, becoming something slightly different than you were yesterday. Every experience leaves its mark, every moment shapes you, every interaction rewires you in subtle ways. Your brain is a universe unto itself, containing multitudes, holding memories spanning decades, skills acquired over years, knowledge accumulated across a lifetime, all compressed into this 3-pound mass of living tissue. And you've only begun to explore its possibilities, only scratched the surface of what this remarkable organ can do, what you can become, what mysteries still wait to be discovered in the intricate folds of your own consciousness. Rest now, dear listener. Let go of the day's concerns. Release the tensions and worries. Allow your muscles to relax completely, feeling your body becoming soft and heavy and comfortable. Let your brain do what it does best, work its quiet magic while you dream. In sleep, your brain processes, consolidates, creates, heals, restores. Trust it to do this work. You don't need to supervise, don't need to try, don't need to do anything but let go and drift, surrendering to the natural rhythms of sleep that have sustained life for hundreds of millions of years of evolution. Your brain knows what to do. It's been perfecting these processes since before our species even existed, refining the art of sleep across countless generations. Let it work. Let it dream. Let it restore you. Tomorrow you'll wake refreshed, renewed, slightly different than you were today, shaped by sleep's transformations, ready to experience the world with fresh eyes and an updated brain. But for now, just rest, just breathe, just be. Your 100 billion neurons will continue their dance, their silent symphony of signals, their endless work of maintaining the mystery and miracle of consciousness itself. Sleep well, knowing that this most complex object in the known universe, your brain, watches over you even as you lose awareness of it, continuing its essential functions, generating dreams, consolidating memories, clearing toxins, preparing you for whatever tomorrow brings. If you enjoyed this journey through the wonders of the human brain, this gentle exploration of neuroscience, this calming dive into the mechanics of consciousness, please like this video and subscribe to Science Lullaby for more soothing explorations of the universe of science. We'll continue exploring the mysteries of existence together, one relaxing video at a time, helping you drift to sleep while expanding your mind. Sweet dreams, dear listener. May your neurons fire peacefully through the night. May your synapses strengthen the memories worth keeping. May your dreams be vivid and strange and wonderful. May your sleep be deep and restorative. And may you wake tomorrow with a renewed appreciation for the extraordinary 3-pound universe residing in your skull, that magnificent brain that makes you uniquely, wonderfully, mysteriously you. Good night. Sleep well. Let science soothe you into slumber.